Appalachian Harvest is a food hub located in Southwest Virginia that provides regional herb and vegetable farmers with training, technical support, aggregation, and distribution services, connecting them to retail markets. We currently source produce from farmers in Virginia, Tennessee, West Virginia, Kentucky, and North Carolina. This webinar is made possible by Risk Management Agency 2018 Risk Management Education Partnership Program.
Welcome to the plant disease section of risk management. And today, as we start to talk about some plant diseases, one of the things we need to understand is exactly what a disease is, how it occurs. And one of the things that we can use for that is what's called a disease triangle. It's sort of a way of thinking about plant diseases in a way that it's an interaction between three different factors, or those three corners of the triangle. You have a susceptible plant, you have a pathogen, which is the disease-causing organism, and then you have favorable environment. If all these factors are present, you will find that a disease will result. But if we can keep one of these factors from occurring, then we will not have a disease. So when we look at methods of disease control, and these are things we'll discuss a little later in greater detail, it can be thought of as we are modifying one of the three corners of the triangle. For example, if we use a resistant variety, let's say a powdery mildew resistant variety, then we are eliminating the susceptible host. And that's going to reduce or even prevent plant disease. Similarly, for some of these diseases, by removing disease plant material, we reduce or eliminate disease because we are removing the pathogen from that disease triangle. And finally, if we can either reduce or eliminate a favorable environment for the disease by doing something as simple as maybe not overwatering and causing high humidity in the plant canopy or the foliage in the garden, then we can prevent that disease from occurring. Again, if we look at this, it takes all three factors. So if we remove one, then there will be no disease present. So what are the causal organisms of these plant diseases? We can divide them into two different categories, one being biotic and the other abiotic. Biotic causes of plant diseases are those that are biological in nature. And we have five major biotic causes of plant diseases. These can be fungi, bacteria, viruses, nematodes, and phytoplasms. Abiotic causes of plant diseases are non-biological in origin. Examples of these would be things such as adverse weather conditions or environmental pollutants. Often what we find is the effects of abiotic factors on plants we refer to more commonly as plant injuries. We'll now proceed to discuss in more detail each of these five biotic causes of plant diseases, as well as some of the abiotic factors also. When we look at the plant diseases, fungi are going to be our largest pathogen group. There are over 200,000 known species of fungi and over 8,000 species which cause plant disease. Now note that the majority of these fungi are not plant pathogens. These fungi primarily survive on non-living organic materials, and we call them saprophytes. But many of these fungi, including some of the plant pathogens, can actually be seen by the naked eye. Typically, however, you're not going to see the major body or the body of the fungus. What we're going to see is simply a reproductive structure of that fungus, and think of that as a mushroom. The non-reproductive body of most fungi, called the vegetative phase, is composed of a mass of fungal threads called hyphae, and the mass of hyphae together is called mycelium. And while an individual hyphae is microscopic, oftentimes mycelia are visible with the naked eye. Fungi reproduce by producing spores that can be in a variety of different shapes or colors. They're produced in a wide variety of reproductive structures that in general we refer to as fruiting bodies. And most of these fungal plant pathogens, except for the few that produce mushrooms, form fruiting bodies that are microscopic in size. Because the characteristics of fungal spores and fruiting bodies are what help us to identify fungi, we can accurately diagnose fungal diseases if we have a microscope. If we do not have a microscope, being able to identify exactly which one it is can be difficult, if not impossible. Looking underneath the microscope at one of these plants that have been infected by a fungi, we find this single thread-like structure called the hyphae that comprise the major body of the fungus. We can think of the hyphae as being a long tube. The more fungi that produce hyphae, what we find is that these hyphae then they cross the walls or these partitions dividing the tubes into individual cells. Now, one of the things that we can note is the presence or absence of cross walls, such as in the lower right-hand corner. This is one of the things that can be a distinguishing characteristics to identify some of the plant pathogenic fungi. The location of the cross wall can also be diagnostic for us as well. In this example, this cross wall's at a branch in the hyphae, and they're offset just a tiny bit, and we can look at the arrow there to see that offset. When we see that, it does give sort of a T-shaped look to it. This configuration of cross walls we can identify as being characteristics of the fungus Rhizoctonia, which is one of the common root rot fungi. We also can look at the asexual reproductive structures of these fungi, and this is what we call the fruiting bodies. The small black blobs that we see in this photo are fruiting bodies. These in particular are pycnidia of the fungus Septoria, which can cause spots on a variety of different plants, including the plant on this slide, which is a cucumber. Within the fruiting bodies, the fungus is going to produce spores. And some of these fruiting bodies we know are asexual in nature. The spores produced in these fruiting bodies are not the result of a male and a female or recombination of genes from more than one fungal parent. These spores are produced by a single fungal parent, and thus when these spores germinate, they produce offspring genetically identical to their parent fungus. We can also look at sexual reproductive structures of the fungi. And what we see with these is the fruiting body, where the black arrow is pointing to that sexual fruiting body of the powdery mildew fungus called Microsphaeria. The spore that we see with the blue arrow pointing is the sexual fruiting body that has resulted from the recombination of genes from two fungal, for lack of better terms, parents. When these spores germinate, they produce offspring that are genetically different from both parents. Now, because this gives it a genetic diversification, a lot of times this means that these are the types of pathogens that will adapt to the products that we use for control, and we can find resistance a little bit more quickly. Now notice that there's a couple of interesting characteristics on this powdery mildew spore, and that is that it's producing sacs, if you see where the red arrow is pointing, and they have these arm-like attachments. If you see the green one, the green arrow, those are called appendages, and the number of sacs of spores are more than one on here, and we see the shape of the appendages that sort of look like antlers. This is the characteristic that we use to classify powdery mildew fungus into the appropriate genus. Now, when we look at the spores itself, these can be interesting as we look at the identifying characteristics that the spores have among these fruiting bodies also. Fungal spores in some ways can be thought of as the seeds of the fungus, although spores are much less complex than what a true seed would be. These fungal spores come in all different shapes and sizes, and some, like these spores of the fungus Fusarium, are multicellular, where they have more than one cell and are colorless. The canoe or banana shape of these cells is also characteristic to the Fusarium fungus. When I look at Fusarium, I sort of look at it as the Cheshire cat fungus, because the spores remind me of the smile of that Cheshire cat's grin in Alice in Wonderland. We can also look at other fungi, such as Theliopsis in this photo, and it produces spores that are again multicellular, but they have a dark pigment. This dark pigmentation is thought to enhance the ability of the spores to survive exposure to adverse environmental conditions, such as exposure to ultraviolet light, and to ward off attack by other parasitic soil fungi and bacteria. So these sort of look like Tootsie Rolls, because the spores that it produces, while they are multicellular, break into individual cells just like the section of a Tootsie Roll would. Spores typically function as a means of reproduction and then as a dispersal for the fungus. They can also serve as a means of survival during periods of unfavorable environmental conditions, and the spores that we see here, called oospores in this photo, are the most common root rot fungus, called Pythium. Now note that these spores have very thick walls that help prevent the loss of water, and they also serve as a barrier to prevent attack by other microorganisms. We've been able to document spores of this type surviving over 40 years in soils. As noted previously, in order for us to accurately identify most fungal plant diseases, we need to look for and then identify the spores or the fruiting bodies that are produced by the pathogen. Some fungal diseases, however, can be identified by eye without the aid of a microscope. One such disease is powdery mildew, that's shown on this photo in a African violet. If you notice the white coating on the top of the surface of the leaf or the flowers of a plant that looks like they've been sprinkled with powdered sugar or talcum powder, then powdery mildew is most likely the suspect problem. The white that you see is a combination of both hyphae and asexual spores. The name powdery mildew actually describes a group of diseases that occur on a wide range of plants and are caused by several closely related fungi. These fungi tend to be relatively related. So what we find is that fungus that would cause the powdery mildew on African violet would not be the same fungus that would cause powdery mildew on, let's say, cucumbers or squash. However, if you see powdery mildew on one plant, look for it on other plants, because it means that the environmental conditions are favorable for it to occur on the other plants as well. Like powdery mildews, rusts, seen in this picture by rose rust, are easily identified by the eye also. This rusty orange pustule, and that's where the arrow is pointing, are produced by rusts and are very distinct and characteristic to the rust. The spores of rust fungi are easily removed from the leaf surface by gently rubbing. The rust fungi are particularly incredible fungi if you look at it. In addition to the typical orange spores that we find, these fungi can produce up to 4 additional distinct spore types. Some of the rusts also have alternate hosts, spending some of their life cycle on one host and then the rest on a second host. The most classic example of this is cedar apple rust, which spends a portion of its life cycle on red cedar and then the rest of its life cycle on apple. Between the two hosts, this rust fungi produces 4 different types of spores. So we're able to identify powdery mildew and rusts in part because the pathogen produces distinct fungal structures. So these are things that we refer to as being signs. These are ones that can be easily identified. Most diseases are not going to be as cooperative as these and take a little more work, at least, to identify them. For most fungal diseases, we have to rely on the symptoms that they exhibit to determine the cause of the disease. The symptoms are not simply the thing that we see, but it's usually an abnormality that we find on this plant that arises during the disease development. Most of the time, symptoms do not definitely tell us the exact disease problem, but often symptoms can give us a sense of whether a disease is caused by a fungus or another type of pathogen. So what type of symptoms do we see being caused by fungal pathogens? Sometimes they're spots. It's on this common leaf that we find, and you'll find these spots that show the areas of leaf tissue that have been invaded by the fungus, and then those areas have died and dropped out. Again, it doesn't definitely tell us which one it is, but it gives us an indication that there is a fungus that is causing the issue. When spots or lesions enlarge and then they merge together so that a large area of the leaf dies off or is necrotic and diseased, then we can call this term a blight. Perhaps the most familiar blight that homeowners would encounter, and even those of us who are in the commercial vegetable, would be on tomatoes, and it's Alternaria leaf blight and Septoria leaf blight. Alternaria is on the left, Septoria is on the right. And these are common in especially high humidity years for tomatoes. It causes browning and loss of leaves from the base of the tomato plant and works its way upward. Sometimes we'll find lesions that occur on the stems of herbaceous plants or on the trunks and branches of woody plants that we call cankers. Often these cankers are shrunken or sunken and discolored, whereas an example in this canker disease on honey locusts, the outer bark layer has totally fallen away. Fungi can also cause a wide variety of rots, and these rots involve the whole destruction or the disintegration of a large portion, if not all, of the plant itself. Typically, affected plant parts become discolored and soft, and common fungal roots such as crown rots, shown in this photo, where the crown of the plant is affected, and root rots, where the root tissue is infected and degrade. The disease shown in this picture is southern blight caused by the fungus Sclerotium rolfsii. This disease is one that's traditionally been associated with warmer climates, and the fungus is not thought to survive harsh winters. But one of the things that we have noticed is as the winters are not as cold anymore, we are beginning to see more of this disease build up and becoming more of an issue on tomatoes and pepper plants. One of the things that we can also notice is what is embedded within the plant itself in the mycelia. You can find small tannish to reddish seed-like objects, and the white arrow is pointing to one of those, that are called sclerotia. These are masses of tightly packed hyphae that serve as a resting or as a survival structure for that fungus. Now, fruit rots are another type of rot that's associated with fungi also. Often these rots start off as a dry rot, but then as the fungus invades the tissue and more microorganisms, and in particular bacteria, follow behind the fungi, then we see these secondary microorganisms leading to a soft, slimy, and sort of disgusting symptom that most people think typically with rots. If we look at this particular slide, we're beginning to see Phytophthora fruit rot on yellow squash. Wilts are another common symptom associated with fungal infections, and wilts basically indicate the plant is not getting enough water. So the leaves, or oftentimes the entire plant, will lose its turgidity, and the plant will have a droopy or a wilted appearance to it. And when we see a plant like that, the first thing we think is, well, the plant's not getting enough water. And if we overwater, then we contribute to the pathogen. And in most cases, we're dealing with an issue with a root rot. This particular one is the root rot fungus Pythium, and the root system has been destroyed and is decayed to the point that it can no longer absorb enough water to support this plant's growth. Finally, it could be an issue called a vascular wilt. Vascular wilt pathogens invade the plant's water conductive tissue called xylem and then cause blockages. And once it blocks those cells off, it prohibits any of the water from being able to translocate through that plant. It reduces the water moving from the roots on upward to the stem, and it can contribute to wilt symptoms also. If you suspect the plant has a vascular wilt, then other symptoms that we can look for is called vascular discoloration or vascular browning. As a reaction to this infection, the xylem changes from its normal whitish green color to a brown or green to gray color. And we can find the arrow pointing to that brownish discoloration in the xylem tissue. Now, not all vascular wilts are going to lead to this discoloration, but when it does, it can be an important clue as to the cause of this problem. And this one in particular on maple tree is the fungus verticillium. Now, bacteria are arguably the second most important groups of plant pathogens. There's approximately 200 pathogenic species that we find with bacteria. Now, unlike fungi, bacteria are not visible to the naked eye, and they have to be looked at using a microscope. Now, for the most part, bacteria are also extremely simple. They are unicellular or single-celled, although certain bacteria have cells arranged in structures that are physically but not functionally similar to a fungal hyphae. Reproductive speaking, bacteria are also simple. They do not form complex fruiting bodies, but they produce by the process of binary fission. So in this process, the existing cells enlarge, and then they lyse or what's divide in half. So these two cells result from the process and are genetically identical to each other and of the parent cell where they have arisen. Bacterial cells, as we notice the arrow pointing to, are extremely small, even when magnified to 400 times their normal size, as they are in this photo. They're difficult to distinguish as individual objects. So a lot of times we'll have to use a stain to stain the cells for us to be able to see them. In general, bacterial cells can be either spherical or round in shape or long and rod shaped. Most of our plant-based bacteria produce rod-shaped cells. When we see these bacterial diseases, this is what produces a lot of our spot symptoms. Often these spots produced by bacteria will have an initial water-soaked appearance with a well-defined yellow halo. But then we need to be cautious when we attempt to distinguish bacterial and fungal spots. Spots produced by bacteria and fungi typically can be very similar, especially in the early stages. Another characteristic that can somewhat be useful to distinguish bacterial spots from fungal spots is their shape. Bacterial lesions are often limited by the veins, or they stay within the border of the veins and have sort of an angular type appearance. Fungal lesions can also be limited by the veins, but more often they're not and have irregular, more round shapes to them. If you suspect that a spot or a lesion has been caused by bacterial infection and you have access to a microscope, then you can confirm this suspicion by looking for what we call bacterial streaming. We can cut the lesion from the leaf or the stem, place it on a microscope slide within a drop of water, and then slice through that lesion with a razor blade. Looking under the microscope, what we will find is the bacteria will stream out of the cut edges of the lesion. In this photo, the grainy material that we see are bacterial cells that are streaming from the lesion that has been cut. When bacterial diseases affect large sections of the entire leaf or the plant, we term this a blight. This photo shows bacterial blight of geranium that's caused by the bacterium Xanthomonas. And because bacterial pathogens tend to be restricted in their movement by veins, often blighted areas on leaves caused by bacteria will have a wedge type shape. The widest area of the wedge is going to be at the edge of the leaf, while the narrowest is going to be near the base of the stem or the petiole. Like fungi, bacteria can also cause rots, as seen with this soft rot of potato. The bacterium Erwinia that causes this disease produces an enzyme that degrades the pectin, or the substance that serves as the glue that holds plant cells together. Once the pectin is degraded, the potato tuber then becomes squishy and turns into that disgusting mess that has that distinct foul smell that we call a rotten potato. We also can look at several classic vascular wilts that are caused by bacterial pathogens. The primest example that we have of bacterial wilt in cucumbers is going to be Verticillium wilt. And if we look at this, we can find that the disease invades, again, the water conducting tissue or the xylem of this plant, plugging that water pathway and leads to a blockage which dehydrates the plant, giving it that wilted look and then leading into death. Sometimes the bacterial wilt of cucurbits can be easily diagnosed in the field. If we cut that wilted vine near the crown of the plant, hold those two pieces apart for a couple of seconds, rub the cut ends together, and then slowly pull the two pieces apart. If we have a bacterial wilt pathogen present, sometimes you will find that slimy strand, almost looking like spider silks, stretching between the two vine pieces. Another symptom that we associate with bacterial infection is the production of galls. And galls are composed of plant tissue where there's an abnormal overproduction of cells, where the cells typically grow abnormally large. While some fungi can cause galls, the classic gall disease, or a crown gall that we see here, is caused by bacteria. This bacterium induces gall formation by injecting a portion of its own DNA into the plant cells, and then the bacterial DNA hijacks the cell's metabolism, and the gall formation is what results. Viruses are also another very common and important cause of plant diseases. And while bacteria are small, viruses are even smaller than bacteria. Viruses are much simpler in the structure than a bacteria also. Bacteria will have what we call true cells. Viruses do not. Most viruses are composed of a piece of genetic material that is within a protein coat. So the viral genetic material can either be DNA, or the same type of genetic material found in human cells, or it can be RNA. Most plants' viruses contain this RNA. And because they're so simple, they don't have the machinery that's necessary to reproduce. So after they infect a cell, they hijack the cell and force it to reproduce its viral particles. Viruses can be transmitted in several different ways. They can be mechanically transmitted either on machinery or our hands. They can also be transmitted by insect, and these insects are what we call vectors. These viruses are picked up by the insect as they feed, removed from the plant, and then injected into the next plant that they feed on. As we talked about before, viruses can only really be seen under special microscopes known as electron microscopes because they're much smaller than even bacteria. This picture shows an electron micrograph with the viral particles magnified about 100,000 times. If we look at it, these particles are spherical in shape, and these plant viruses we know can either be rigid or they can be flexible. Not all viral infections can be necessarily bad, at least from the human standpoint. In some fact, some of these viral infections are desirable. If we look at this tulip, we can find these interesting flower colorings that's been caused by a virus. The classic symptoms that we see from viral infections is called a mosaic. It's the production of the blotchy light and dark green coloring that we see within the foliage. Viruses can also give us interesting patterns within the leaves, and this shows the typical symptoms that we would see on a rose mosaic virus. When these blotchy colorings occur on flowers, the symptom is referred to as a mottle rather than a mosaic. We can also find fruit and leaf distortions with bacteria as well, with viruses as well. This photo shows the leaves and the fruit of zucchini that has a viral infection. Now, if we look at it, these leaves are what we would refer to as being strappy, and they're also curled and twisted. We commonly see these leaves being much thicker, more leathery than normal, and then on the fruit itself, it exhibits a mosaic or a mottled appearance, and almost has these bumps that we would call warts that would come on the fruit also. These are all what we would call typical symptoms of a viral infection. Some viruses can induce a production of concentric ring patterns on the fruit or on the leaves. These are what we refer to as ring spot, and many viruses are going to carry the word ring spot in their name, such as papaya ring spot that's shown here in this photo. In the previous example, ring spot symptoms were simply the discoloration of the fruit tissue and other plant tissues. We don't really see any death of the tissue itself. But in other viral infections, such as this one found in the impatient necrotic spot virus, we can see both the ring spot, but then we also see the death or necrosis of that tissue also. The next grouping that are less likely to be seen are called phytoplasms, and these are what we also call a mycoplasma-like organism, because they sort of resemble an organism that causes a disease in animals. Recently, the name phytoplasm was coined to describe these mycoplasms in plants. They're sort of an odd bacteria-like organism. They're different from true bacteria in that they're smaller and they don't have any type of cell wall. So when we look at the disease that's caused by phytoplasms, we always sort of lump them into the group of viral diseases because that's what they look like. But now we know that phytoplasms live and reproduce in the plant's food-conducting tissue called phloem. As is the case with bacteria, phytoplasms are produced by binary fission, so they enlarge and split in two, and they're transmitted by insects, and in particular, transmitted by the insect leafhoppers. When we look for the signs of phytoplasms, again, they're much smaller than an ordinary bacteria. So like viruses, you have to use a scanning electron microscope to be able to determine whether they are. Now, in the phytoplasms, one of the things about them is they can actually be seen under light microscopy, but it requires a type of fluorescent dye used to use this light microscope. So these dyes actually bind to the DNA, which is the genetic material that the phytoplasms possess, and then they glow when they're exposed to ultraviolet light. The photo here shows phytoplasm cells that's contained within a phloem cell that have been stained and then exposed to ultraviolet light. The symptoms that we associate with phytoplasma infections are sort of the odd and unique ones. One of the most common symptoms is what we call virescence. It's a normally colored plant part, such as a flower, that are green in color. Then we can also find other common symptoms called brooming or witch's brooms, where the plant parts becomes overly branched and bushy. The coneflower on the right of this flower shows both of those symptoms. The petals are short and stumpy, have a green appearance, and we also see the brooming effect of the flower, the seed head portion itself. Brooming is also common among trees and other type of things. And the example that we see here is an extremely severe sample of the symptoms of brooming. Another symptom that we see, and in particular we find these with winter and summer squashes, are fasciations. And this is where the flattening of the plant parts occur. Normally this is on the ends, or what we call the terminal ends of the vines as they're growing. And again, they flatten out and they grow wide, extremely thick and tough. These cosmos are suffering from what's called Astor's yellows, and they're showing the typical virescent symptoms. We see also the twisting and the distortion of the plant that can also be characteristic with a phytoplasma infection. Now, nematodes represent the final major groups of what we call plant pathogens, and these organisms rival, if not exceed, fungi in their complexity. Nematodes are very large when we compare them to other pathogens and are readily seen with a light microscope and on occasion with the naked eye. They have a sensory, digestive, and reproductive organ, and these nematodes reproduce through the production of eggs and go through several juvenile phases before they become adults. When we look at nematodes, they're easily identified once they are extracted from the soil or from infected plant material. The biggest issue that we have with nematodes is being able to distinguish what is either a plant pathogenic or a non-plant pathogenic nematode. Typically, non-pathogenic nematodes are going to be much higher in concentration than we find the pathogenic nematodes in a mix. So in order to distinguish the two, we look for a stylet, which is the piercing mouth part of the pathogenic nematode. The classic symptom associated with an infection of nematodes is the formation of these root galls. The root knot nematode will cause these types of symptoms. The nematodes tunnel into the root, and that's where it sets up and starts to reproduce. And as that nematode feeds, the saliva stimulates the plant root cells to divide excessively and then to expand to larger than their normal size. This leads to this large knot or gall formation. Now, while most nematodes cause a root symptom either as a gall or as general tissue necrosis, there are a few nematodes that can cause foliar symptoms also. These are much less common, especially in the vegetable production industry. Next, we'll look at the abiotic factors, which include things such as nutritional abnormalities, pesticide exposure, environmental pollutants, and then reactions to adverse weather conditions. Often nutrient abnormalities show up as discolorations of the foliage. These common discolorations include yellowing or chlorosis or a reddening of the tissue. Note the pattern of yellowing or other type of discoloration can provide us with clues as to which type of abnormality that we are looking at. In this case, the yellowing has occurred along the margins of the leaf, and it's actually due to a lack of the micronutrient molybdenum. General yellowing of foliages can also indicate a lack of nitrogen. If the leaf veins remain green and the leaf blade turns yellow, then we might suspect an iron or a manganese deficiency could be the problem. Reddening often indicates a phosphorus deficiency. While yellowing is often a symptom of a nutrient deficiency, it can also indicate an overabundance of a particular nutrient. If we look at this plant, it actually has received too much copper, which has turned toxic in the plant and has led to the chlorosis that we see. The other issue we deal with in organic gardening, especially if we're close to conventional farms, is that herbicides, when they are used, and even those of us that may be close to homeowners that use pesticides on their lawn, they can drift and we can find accidental exposure onto non-target plants. Now, common symptoms that we normally see include cupping and curling. Tomatoes, as well as grapes, are particularly sensitive to low levels of some herbicides. Another common symptom we see is the twisting of the terminal ends, as noted on this tomato plant. Distortion of flowers is also another common symptom with herbicide exposure. And note that the symptoms of herbicide exposure are often very similar to symptoms caused by either a virus or a phytoplasm. To distinguish between these two can sometimes be very difficult, and those plants need to be sent off for diagnostics to rule out any pathogen before we say that it would be an herbicide damage. In addition to improper use of herbicides, improper uses of fungicides and some insecticides can lead to plant injury. For example, in this one, we find marginal burning of the leaf because of an application 4 times the labeled rate of a common root rot fungicide. So when we use pesticides, and yes, that includes organic pesticides, make sure that you read and follow all of the instructions on the label, including the recommended rate. In the state of Virginia, we have to understand that the label is the law. It is a binding contract between the company, the EPA, and you as the applicator. If you go off that labeled rate and cause injury, you have voided your warranty with them. Another thing we might see is contaminants in water or the air that affect the plant health. These pollutants can cause a wide range of symptoms, many of which look like damages caused either by nutritional abnormalities or even insect damage. In this photo, we see marginal bronzing due to an exposure to sulfur dioxide. Sometimes we will see these high levels of sulfur dioxide in greenhouse plants that are being grown, especially where they're using coal in the stokers to heat those greenhouses. High levels of fluoride in water or in the air can also cause symptoms like this seen. Normally what we will find is papery thin, almost transparent looking lesions that we would normally think would come from either a fungi or a bacteria. But we also can see here that the damage is concentrated toward the margin of the leaves, and this is where fluoride tends to accumulate. Now, some environmental pollutants can have biological origins. One of the most interesting ones that I have worked over the years is when a person called me out to look at a plant in their house. The bottom leaves were being burned off. They could not figure out what it was. When I made the house visit, the door opened. The family dog came out of the house and he hiked his leg on the bush. So we see the damage here due to excess nitrates from dog urine. Other times we may look at plants and think that there's been some type of catastrophic event. Either a neighbor has sprayed an herbicide that has drifted and has caused death, but also extreme weather conditions are to play with plant injuries also, especially when we have these cold snaps and we have tender plants. If we look here, these new branch tips or the candles on Christmas trees have been burned by low temperatures and extreme frost and freeze that occurred while they were susceptible. On the other hand, we can also see symptoms due to heat injuries. This is actually a ginseng garden, and the temperature of the heat got high enough that it caused tissue damage to these ginseng plants. One of the things that helps us with this is knowing what the weather conditions were prior to the onset of the symptoms that we see. And sometimes that's the only thing that leads to the identification of these environmental factors. Often lack of water causes some injury to plants, but in this photo we see the disorder caused by winter injury. This was where we had a layer of water that was on the plant itself. As the temperatures began to rise, the foliage of the shrub became actively growing. It began to use the water, and when it did not uh, grow enough, what we found is that these leaves actually started to die because there was not enough water being pulled up by the roots, which hadn't got warm enough to be activated, and the translocation of that water being taken up. So the leaves started growing because of the air temperature. The roots did not activate because of the soil temperature, and we see just an abnormality caused by two separate temperatures. We can also see issues during times of drought. When we don't have the rain that's plentiful enough, we can see symptoms of drought that can manifest as discolorations on plants that may be typical of other type of fungi or bacterial infections also. So far we've discussed biotic and abiotic causes of plant diseases. In this next session, we're going to discuss some methods for disease control. And we're going to look at cultural control. This is the most common way that we may attempt to control plant diseases. And one of the things that I look at on cultural control is it is modifying how you grow your plants in order to reduce the environment for disease growth. We can also try to avoid the pathogen. There's several straightforward ways that we can do that, and one of them is buying seed from reputable sources. Many of our plant pathogens, particularly viruses and bacteria, are carried on seeds. So we have to choose clean seed from reputable sources to minimize those disease problems later on. We also want to inspect our plants prior to our purchasing if we buy transplants. Make sure that the plants look healthy. If they're obviously diseased or look stunted, don't buy them. In addition to looking at the leaves and the stems and the branches, try to get a look at the plant's root system. If we see an overabundance of brown roots, that might be an indication of a root rot or a nematode problem. We also want to inspect for any insect pests that could cause some serious damage to the plants through their feeding. Again, if we have excess insect feeding, it's going to reduce that plant's ability to fight off disease, and it's going to be in a weakened state. We may want to control alternate hosts, things that may carry a particular disease, such as in the case of cedar apple rust. We may want to cut the cedar trees down. Of course, most of us aren't going to have the ability to cut cedars between a quarter and a half a mile away. The other thing is to use non-contaminated water. Sometimes the water that we use has come into contact with soil or plant pathogens, and it can infect the plant material if we use it as a source of our water. When we look at cultural controls, we want to avoid environmental conditions that favor disease. Notice that we spoke about a favorable environment as one of the three sides of the disease triangle. So if we eliminate as much of the environment as we can, we're going to either eliminate or at least dramatically reduce the disease that we can see. Now, sometimes we may not have what we would consider to be much control over the environmental conditions, but we can at least take measures. We want to make sure that we maintain a moderate soil moisture. We don't want to overwater, increasing the amount of moisture and humidity. If we're growing in a greenhouse, we want to reduce the humidity within the greenhouse. We want to fertilize our plants properly. By having a balanced nutrition, it impacts that plant's health. Plants with nutrient deficiencies tend to be more susceptible to infection than those with proper nutrient balance. But we also don't want to over fertilize, particularly with nitrogen, because excess nitrogen can tend to produce a fast growing, very succulent growth with tissue that's more susceptible to infection by certain pathogens. We want to prevent water and heat stress. Again, if we look at those plants, if we stress them, then they are more likely to become diseased than healthy plants. And then be aware of activities surrounding those plants. Are there any type of environmental pollutants that might be close that could cause issues? And if there are, we need to take actions to minimize the impact of these factors. We also want to look at sanitation. We want to disinfest our tools. So if we are using gardening tools, especially around plants that we know might harbor a virus, we want to disinfect those tools before we use them on another plant. We can use sodium hypochlorite, alcohol, or other types of approved disinfectants to do that. Some folks have asked whether Lysol will work, and it should not be used in most cases because it is not labeled for that particular use. We want to clean pots and benches if we're using greenhouses. Make sure that all of our worktops are washed and thoroughly disinfected. Use pasteurized soil. If we're using common compost, we have to understand that there's a lot of microorganisms that have broken that soil down. But once that occurs, then it also sets up the perfect place for plant pathogens to also grow. So if we can use a pasteurized soil, that means it's been heat treated to the point that all the pathogens have been killed, and we're going to reduce introducing those diseases to those plants. If we're using recycled water, we want to make sure that we're decontaminating it, either filtering or running it through a UV radiation treatment to make sure that it's safe to go back on those plants. If we do find diseased plants, we want to remove those as quickly as possible so that it's not a source of inoculum to other plants. And then if we find dying plants, we want to remove them also. Again, if we have those dying plants, we know that there are certain bacteria that will come in after those plants are beginning to die, and they become a place for those bacteria and some of our fungi to set up, and can be what I call the typhoid Mary, the one that continues to produce the disease to infect other plants. Now, one of the other things we can look at after everything else has been utilized is looking at chemical control. So we do have fungicides, which control fungi, bactericides, which control bacteria, and nematicides, which control nematodes. And there are organic versions of all of those. Your white mustards can be used for nematode control. And when we look at these fungicides and bactericides, for most of us in the organics, they're going to fall into the protectant category. Protectant fungicides are ones that have to be applied to the surface of a plant, and it has to coat that surface of the plant to be effective. The protectant fungicides are not absorbed by the plant, so basically all they do is form a chemical barrier between the pathogen and the plant. We have to have good coverage for those protectant fungicides to be effective, and we have to reapply at labeled rates to make sure that we're maximizing the effectiveness of that protective fungicide. When we look at any product that we use, we need to make sure that it is registered and it is labeled for the crop and the condition in which we are growing. The other thing is, just because a pesticide is labeled organic does not mean it is non-toxic. Remember, it has to kill something, which means it has some type of deadly characteristics to it. Because of that, you want to make sure that you use products safely. Make sure that you're using gloves. Wear long pants, long sleeve shirt. Wear a pair of protective glasses. That way you can minimize your exposure to those products. Then once you're done, go inside, wash those clothing, and take a shower to make sure you're getting those chemicals off. The other thing that we highly suggest is going through a pesticide applicator training. One of the things that it does is it gives you the knowledge that you need to know how to handle pesticides properly. Now, looking more into some of the biological controls, the biggest one that we can look at is disease-resistant plants, those that have a natural resistance to certain diseases, and that gives us an opportunity to fight it before the disease ever shows up. The other thing that we can look at is using biological pesticides, and those can be plant extracts or antagonistic microorganisms. These biological pesticides are similar in many ways to the inorganic chemical pesticides. They're applied to the plant in order to control a plant disease, but they originate from something biological. These pesticides can often be plant extracts that inhibit the growth of the reproduction of that plant pathogen. But most of the time, we find that these biological pesticides are composed of microorganisms, other bacteria or fungi, that either compete with the pathogen to prevent or at least reduce the amount of disease that we see. Now, diagnosing plant diseases is not going to be an easy task. But one of the things that you can do is consult with your local county extension agent or your extension specialists and allow them to help get the ball started on identifying some of these plants. If not, each of the universities have disease labs where we can send specimens in to identify exactly what it is we're dealing with, and we can assist you in being able to identify the disease and hopefully come up with a control measure that will be satisfactory for your production. If you have any questions, my contact information is listed here, both my email and my office phone number.